Welcome back to the Deep Dive. Today, we are really undertaking something quite ambitious. A mission of mastery, you could say.
We are.
We're charting what has to be one of the most complex and, well, definitely the most precise chronological event in human biology.
Absolutely.
We're diving deep, really deep, into the first 2 weeks of life.
Yeah.
It's a magnificent biological feat when you think about it. 2 highly specialized single cells transforming into this multi-layered structure that actually implants itself in less than 2 weeks.
It is truly foundational knowledge, and today we're aiming to go beyond just a simple summary. We're using, dense textbook material as our guide here to really explore the cellular and the hormonal choreography that's absolutely necessary for reproduction. We need to understand not just what happens, but crucially, why it happens with such, staggering precision.
Exactly.
So our goal today is for you, listening, to really master this timeline and the mechanisms that drive it.
Okay, let's unpack this epic journey then. Starting right at the beginning, gametogenesis. This is how those specialized sex cells, the ovum and the spermatozoon, are actually made.
Right.
And the contrast between the female and male approach is, well, it's dramatic.
Gametogenesis, yeah, it's all about specialization, really, and chromosome reduction. Taking the cell from the full diploid set, the 46 chromosomes, down to the haploid number 23.
Half the set.
Exactly. But the timelines for oogenesis, which is the female process, and spermatogenesis, the male process, they are just worlds apart. Really couldn't be more different.
Let's start with oogenesis then. The timeline is, frankly, almost unthinkable when you lay it out.
Yeah.
We're talking about the primitive germ cells beginning their existence outside the future ovary.
That's right, yeah. They originate from the yolk sac. This is way back around the end of the 3rd week of gestation.
Third week.
And then they migrate to the developing gonadal ridge, completing that journey by the end of the 4th week.
So they have to travel even before they start dividing properly.
They do. And once they arrive, they differentiate into oogogonia. And this happens via really rapid mitotic divisions, just copying themselves.
Okay.
But the numbers are astronomical and kind of terrifyingly finite. The absolute peak count of oogogonia is reached around the 20th week of gestation.
20th week, so halfway through pregnancy?
Pretty much. And the number is about 7 million.
7 million potential eggs.
That's the maximum potential reproductive capital a female will ever possess. Ever.
And the clock immediately starts ticking down from that point.
Yeah.
What happens next really defines this incredible biological commitment of the female germline, doesn't it?
It really does. While some oogogonia continue dividing for a bit, others, they start something different. They enter the prophys of the first meiotic division.
Meiosis, the reduction division.
Exactly. And as soon as they do, they become primary oocytes. And they're immediately packaged up, surrounded by these flat cells, forming primordial follicles right there in the cortex of the ovary.
So they're already preparing for a potential future ovulation, decades down the line.
In a way, yes. But the crucial step, the defining step, is the arrest. At birth, all the oogonia are gone. They've all been replaced by these primary oocytes.
All 2 million or so.
Roughly 2 million, yes. And these cells enter a resting phase. It's called the dictyotene stage. They're basically suspended, frozen in time, between the prophase and metaphase of that first meiotic division.
Okay, let's pause on the dictyotene stage for just a second, because this is where the biological risk, the vulnerability, truly lies, doesn't it? This means the primary oocyte is literally the longest-lived cell in the human body.
It is.
Its delicate genetic material is held hostage, essentially, suspended in time for potentially decades, vulnerable to environmental damage, mutations, just aging.
It's a massive biological gamble, a huge investment. And the attrition, the loss, it continues relentlessly. That 2 million count at birth is swiftly, drastically reduced.
How much reduced?
By puberty, estimates suggest only about 400,000 primary oocytes remain.
From 2 million down to 400,000?
Yes. This selection is just brutal. And then, get this, out of those 400,000, only approximately 400, maybe 450, are ever likely to actually ovulate during the entire reproductive period.
400 out of an initial 7 million.
Exactly, that ratio.
Yeah.
It just highlights the intense, almost unbelievable selectivity of the reproductive process.
So if the whole point of maturation is chromosome reduction, getting to that haploid number, how does that finally happen after decades of waiting in this arrested state?
Okay, so just before the first meiotic division is finally allowed to complete, the DNA actually doubles by replication. So the primary oocyte still has its 46 chromosomes technically, but each is doubled. 22 pairs of autosomes and 1 pair of sex chromosomes, which are XX in females.
Right.
Then the first meiotic division is triggered, and this happens just prior to ovulation, and this is critical.
Yep.
It results in 2 unequal cells. Very unequal.
Why must the division be so unequal? Why sort of sacrifice cytoplasm from one of the resulting cells?
It's all about resource allocation. That sacrifice ensures that the future ovum receives virtually all the necessary cellular machinery, the mitochondria, the ribosomes, the nutrient stores, everything it needs for the first few days if fertilization happens.
Ah, I see. So it concentrates the good stuff.
Exactly. The result is one large secondary oocyte, now haploid, technically 23 chromosomes, each still doubled, so 23X, retaining nearly all the cytoplasm. And then you get this tiny, small first polar body, also 23X, but with hardly any cytoplasm.
And the polar body just degenerates.
It's essentially an efficient way to discard the excess chromosomes without sacrificing those essential cytoplasmic resources. It might divide again, but it plays no further role.
Okay, so that's the first meiotic division done.
Yeah.
But the second meiotic division, that's the final step to get a truly mature ovum, but its completion is conditional, right? It depends on something else happening.
Precisely. The secondary oocyte begins the second meiotic division, but it arrests again. This time in metaphase 2. It's poised, ready to proceed, but only completes this division after fertilization by the sperm.
Only if sperm arrives.
Only if fertilization occurs, typically in the fallopian tube. If it does, then meiosis to second completes, yielding the mature ovum, the larger functional cell, now truly haploid 23X, and a second polar body, which also gets discarded.
And if no fertilization happens?
If there's no fertilization, the secondary oocyte simply degenerates within about 12 to 24 hours. It never completes its final division.
Let's quickly detail the structure of this final mature ovum then. It's the largest cell in the body, you said, about 130 microns in diameter.
That's right, huge in cellular terms. At the center, you have the cytoplasm, packed with nutrients, and the eccentric nucleus. This contains the 23 chromosomes, 23X, and quickly converts to the female pronucleus once the sperm enters.
Okay, and the layers the sperm has to get through.
Surrounding the cell itself is the vitelline membrane, that's the actual cell membrane, or ulemma. Outside that, you have the zona pellucida. This is a crucial layer, a transparent, quite thick, protective outer envelope made of mucoprotein. Importantly, it has tiny channels for transport.
And wasn't there a space between the membrane and the zona?
Yes, exactly. Nested between the vitelline membrane and the zona pellucida is the paravitelline space. It's a narrow gap, and that's where those polar bodies we talked about end up.
Right.
And then finally, externally, covering everything, is the corona radiata. This is a layer, sometimes several layers, of granulosa cells. They're derived from the cumulus oophoris, the cloud of cells that surrounded the oocyte in the follicle, and the oocyte carries these with it when it's ovulated.
Quite a fortress for the sperm to penetrate.
Absolutely. Each layer presents a challenge.
Okay, now let's shift gears completely to the male counterpart. Spermatogenesis. You said this is worlds apart, a continuous, rapid process that begins shortly before puberty.
Yeah, it's a massive operational difference from the finite start-stop female process. Think of it like a factory that's operational pretty much 24/7 from puberty onwards.
Where does this happen?
It starts with primordial germ cells developing into spermatogonia within the walls of the seminiferous tubules in the testes. These spermatogonia divide mitotically to maintain their population, but some differentiate into primary spermatocytes.
And these have the full set of chromosomes.
Yes, the full diploid set, 22 pairs of autosomes and the XY sex chromosomes. Now, these primary spermatocytes enter the first meiotic division, but they stay in prophysae for a surprisingly specific length of time, about 16 days.
16 days just in prophase. Why is that window so rigidly precise? What's the cellular mechanism dictating that specific timeline?
It seems to be dictated by the intricate timing required for the continuous production cycle within the tubules. It ensures a steady output. This first meiotic division, when it finally completes, forms 2 secondary spermatocytes.
And are these equal or unequal?
These are equal in terms of cytoplasm, and they're now haploid, containing either 23X or 23Y chromosomes.
Ah, so the sex determination happens right here.
Effectively, yes.
Yeah.
The potential sex of the offspring is determined by which type of secondary spermatocyte is formed. And critically, this is immediately followed by the second meiotic division, sometimes called the homotypical division. no delay.
Gametes.
That forms 4 spermatids, also equal in size and haploid.
So unlike oogenesis, where one primary oocyte yields just one functional ovum,
Right.
one primary spermatocyte yields 4 potentially functional spermatids.
Exactly. 4 haploid cells result from each primary spermatocyte that starts meiosis. 2 will be 23X and 2 will be 23Y. Sex determination is now fixed in these cells.
But they're not sperm yet, are they? Spermatids.
Correct. They're not yet functional sperm. They're basically round, non-motile cells. They must undergo this extensive morphological differentiation process called spermiogenesis.
Spermiogenesis, so changing shape and function.
Exactly. It's a remarkable transformation. They shed most of their cytoplasm, grow a tail, develop the acrosome cap, and importantly, this happens without any further cell division. They just remodel themselves.
And how long does this whole production line take? From the initial spermatogonium to a fully mature spermatozoon?
The entire process takes a remarkably consistent 61 days, give or take a day or two. It's a very tightly regulated schedule.
Okay, the structure of the mature spermatozoon. It's clearly engineered for delivery, optimized for speed and penetration. We should detail its components.
Definitely. It's defined by its 2 main parts, the head and the tail. The head is primarily composed of the highly condensed nucleus carrying the genetic payload and, crucially, the acrosomal cap.
The acrosome, that's the enzyme pack.
Precisely. It covers the anterior part of the nucleus and is packed with lyditic enzymes like hyaluronidase and acrosin that are absolutely necessary to breach the ovum's outer layers.
And the tail, the propulsion system. That's divided into sections too, isn't it?
Yes, it's quite complex. There's the neck, which connects the head to the tail. Then the middle piece, this is the powerhouse section.
Because of mitochondria.
Exactly. It's packed with mitochondria arranged in a spiral, providing the ATP, the energy needed for the tail's movement. Then you have the principal piece, which is the longest part of the tail, and finally the short end piece.
And the actual mechanism of movement. How does the tail whip?
It's driven by the X-normal complex running down the core of the tail. This is a classic piece of microengineering, the 9+2 structure you see in cilia and flagella elsewhere.
9+2?
If you view it in cross-section, you see 9 pairs, or doublets, of microtubules arranged in a circle around 2 central single microtubules. These peripheral doublets are joined by links, like Nexin links, and crucially, they have these tiny motor proteins called dynein arms attached.
Dynein arms, like little molecular motors.
Exactly. These dynein arms use the ATP generated in the middle piece to walk along the adjacent microtubule doublet. This causes sliding between the doublets, which results in the bending motion, the whip-like action, that propels the sperm forward. It's a biological motor built for speed and endurance.
Okay, so we have our highly specialized cells ready. The secondary oocyte waiting, the spermatozoa produced. Now, we need to orchestrate their meeting. Section 2 is all about ovulation, that critical moment, the release of the secondary oocyte.
Right. It follows the rupture of a mature Graafian follicle, making the oocyte available for conception.
And when does this typically happen?
Usually, it occurs about 14 days prior to the expected start of the next menstrual period. So in a classic 28-day cycle, that's day 14. But the key is it's timed relative to the next period, not the last one.
And it's usually just one oocyte cycle.
Typically, yes. Just one secondary oocyte per ovarian cycle, though twins can happen. It's also worth remembering, as the sources point out, that ovulation can be suspended obviously during pregnancy. And sometimes menstruation can occur without ovulation and ovular cycles.
Oh, right.
These are more common during times when hormonal rhythms are less stable, like during adolescence, just after childbirth, and as women approach menopause.
Okay. Let's discuss the mechanics then. What's happening in the ovary just before release? The preovulatory changes. The Graafian follicle swells up massively, right?
Dramatically. Due to a huge accumulation of follicular fluid, it reaches about 20 millimeters in diameter. It really bulges on the surface of the ovary.
And inside, what's happening with the oocyte and its surrounding cells?
Inside the follicle, that cumulus oophoris, the big cloud of granulosa cells holding the oocyte, actually detaches. It separates from the follicle wall and floats freely in the antrum, the fluid-filled cavity.
So it's ready to leave.
Exactly. And at the same time, the inner layer of those cumulus cells, the ones immediately surrounding the oocyte, arrange themselves radially, sticking out like rays of the sun. This forms the corona radiata that we mentioned earlier. The oocyte is definitely preparing for its exit.
And the rupture point itself. The sources mention the stigma.
Yes, the stigma. It's like a small, conical, weak spot that develops on the follicle wall, penetrating the outer ovarian surface layer. It's very transient, though. It exists only momentarily, maybe half a minute to 2 minutes, just as a thin membrane.
And then it doesn't pop like a balloon.
No, it's less of an explosion and more of a controlled ooze. The cumulus mass containing the oocyte slowly extrudes out along with the follicular fluid. This whole expulsion process takes about 1 to 2 minutes.
That's surprisingly slow.
It is, and once the oocyte is out, the rupture site, the stigma, is quickly sealed by a plug of plasma. Importantly, this physical release is preceded by the final, crucial preparatory steps within the oocyte itself.
Right, the final touches to get it ready. What are those?
Well, there are significant cytoplasmic changes. The volume increases a bit more, and there are changes in the number and distribution of key organelles like mitochondria and the Golgi apparatus, getting everything optimally positioned.
And the nucleus.
Most importantly, this is when the arrested first meiotic division finally completes, prompted by the hormonal surge we'll discuss. This results in the extrusion of that first polar body and the formation of the secondary oocyte, now with its 23X chromosome complement, ready for potential fertilization.
Okay, so what is the engine driving this whole release? The endocrinal control. This must be tightly regulated.
Extremely. The entire event is initiated by the luteinizing hormone surge, the LH surge.
LH, from the pituitary gland.
Yes. And this surge itself is caused by a sustained high level, a peak level, of estrogen that has been produced by the maturing follicle. This estrogen peak needs to last for about 24 to 36 hours in the late follicular phase. It's the duration of high estrogen that flips the switch in the pituitary.
It needs to be sustained to trigger the surge.
Precisely. And the timing from that point is key. Ovulation occurs approximately 16 to 24 hours after the LH surge begins. The LH peak itself persists for about 24 hours, which gives the system a necessary window for all the subsequent actions to occur.
And what does LH actually do at the follicle level?
It's like a molecular command center. First, it stimulates the completion of meiosis in the oocyte, as we just discussed. Second, and crucially, it initiates luteinization of the granulosa cells and the theca cells in the follicle wall.
Luteinization,
Yeah,
becoming the corpus luteum.
Exactly. This transformation starts even before ovulation. It leads to the synthesis of progesterone, which is vital, and also prostaglandins. Both are key players in the rupture process itself, and of course progesterone is essential for maintaining the uterine lining later.
Is LH the only player? What about FSH, follicle-stimulating hormone?
There's an FSH surge too, yes. It happens alongside the LH surge. And it seems to be facilitated by that pre-ovulatory rise of progesterone. That LH initiated an interesting positive feedback loop involving estrogen and then progesterone.
And what's FSH's role in ovulation?
The FSH surge appears to increase the levels of plasminogen activator in the follicle. This converts plasminogen into plasmin.
Plasma. That's an enzyme that breaks down proteins, right?
Exactly. Plasmin is a key player here because it helps in the lysis, the breakdown of the proteins in the follicular wall. It helps weaken the structure for rupture. So you can see the combined mid-cycle LH and FSH surge orchestrates everything. Final oocyte maturation, weakening and rupture of the follicle wall, and the expulsion of the oocyte.
It's like a perfectly timed biological demolition job.
You could say that.
What's fascinating, though, is the point about pressure. We often imagine ovulation as a pressure burst, like popping a blister. But the sources say the intrafollicular pressure actually remains pretty static, around 15 millimeters of mercury.
That's right. It tells us the rupture isn't primarily a hydraulic explosion. It's more driven by enzymatic degradation of the wall, that plasmin activity, and passive stretching as the follicle reaches its maximum size.
A controlled enzymatic melt and collapse, perhaps.
That's a good way to put it. And that passive stretching and weakening is also aided by some mechanical factors. There's evidence for contraction of tiny smooth muscle fibers, micromuscles, found in the theca external layer of the follicle and in the surrounding ovarian stroma.
And what makes them contract?
Those prostaglandins, the ones whose synthesis was kicked off by the LH surge, they stimulate these muscle contractions. So it's a beautiful integration. Hormonal command triggers enzyme release and prostaglandin production, which in turn weaken the wall and cause localized contractions to help push the oocyte out.
Right. The secondary oocyte is released, floating near the ovary. Now, the race for fertilization begins. And the location is almost always the same specific place, isn't it?
Almost always, yes. The ampullary part of the uterine tube, or fallopian tube, the widest part, further away from the uterus.
And how does the oocyte get there? It doesn't just drift randomly.
No, it's actively picked up. The fimbriae, those finger-like projections at the end of the tube, sweep over the ovary surface. This pickup isn't random. It involves coordinated muscular action of the tube, maybe some suction created by fluid currents, definitely the action of cilia lining the tube, and possibly even positive chemotaxis.
Chemotaxis, like chemical attraction.
Yes, there's evidence that secretions from the tube might release chemicals that attract the oocyte cumulus complex towards the opening. Once picked up, it's rapidly transported to the ampulla.
Okay, time is now absolutely critical. What's the window?
Very narrow for the oocyte. Its fertilizable lifespan is estimated to be only about 12 to 24 hours after ovulation.
That's it?
That's it. Sperm, on the other hand, are much hardier. They can survive within the female reproductive tract for maybe 48 to 72 hours.
So the timing of intercourse relative to ovulation is key.
Absolutely critical. And consider the journey the sperm have to make. Hundreds of millions might be deposited in the vagina.
Hundreds of millions.
Yes, but only a tiny fraction make it all the way. Maybe only a few hundred, the sources estimate around 300 to 500, actually reach the vicinity of the ovum in the ampulla.
That's an incredible loss rate.
It is. The journey is long and hazardous. But sperm transport itself can be surprisingly fast. Some can reach the tube within minutes, helped by uterine contractions. But reaching the tube isn't enough. Before they can even think about fertilizing, the sperm must undergo a fundamental transformation.
Right. They need to be activated. This starts with capacitation. What exactly is this physiochemical change, and why is it absolutely mandatory?
Capacitation is essentially a period of conditioning or priming that sperm undergo within the female genital tract. It takes several hours, typically 2 to 6. It involves stripping away certain molecules, glycoproteins and sterols, from the sperm's surface membrane that were acquired in the epididymis.
So removing an inhibitory coating.
In essence, yes. This process unmasks binding sites and makes the membrane more fluid and permeable, particularly to calcium ions. It also involves biochemical changes inside the sperm, things like cyclic AMP-dependent phosphorylation. And there's a crucial shift in the cell's internal ion balance, an influx of calcium ions and an efflux of hydrogen ions.
What does that ion shift do?
It increases the intracellular pH. All these changes together result in two key things. First, the sperm become hypermotile. They switch to a more vigorous, whip-like swimming pattern needed near the egg. Second, and crucially, it enables them to undergo the next step, the acrosome reaction.
Wait, so capacitation isn't just about swimming faster. It's fundamentally rewriting the cell's internal state, its energy management, and enabling its binding capability.
Yeah.
It has to happen in the female tract.
Exactly. It's a cellular switch, flipping the sperm from, say, storage or transport mode into full-on invasion mode. They cannot fertilize without it. Then, when a capacitated sperm encounters the oocyte or its surrounding layers, the next stage is triggered, the acrosome reaction.
An enzyme release.
Yes. The outer membrane of the acrosome fuses with the sperm's plasma membrane at multiple points, creating pores. This allows the explosive release of those essential lytic enzymes stored inside the acrosomal cap. Hyaluronidase, various hydrolytic enzymes, proacrosin, which gets converted to active acrosin.
And these enzymes are the biochemical keys needed to breach the oocyte's defenses, starting with the corona radiata.
Correct. Hyaluronidase, likely released from many sperm in the vicinity, helps to digest the hyaluronic acid matrix holding the corona radiata cells together, allowing sperm to push through that outer layer.
Okay, corona cells dispersed. Next is the zona pellucida. This seems tougher.
It is. Penetration of the zona is facilitated primarily by acrosin, which digests the zona proteins. But the binding interaction here is fascinatingly specific. The sources highlight a two-step process. First, a sperm with an intact acrosome initially binds to a specific zona protein called zona protein 3, or ZP3.
ZP3 binding triggers the acrosome reaction.
It's considered the primary trigger for the acrosome reaction in vivo, yes. After the acrosome reaction has occurred, the sperm then binds to a different zona protein 2, or ZP2. This secondary binding helps anchor the sperm as it uses its enzymes and motility to digest its way through the zona.
That molecular precision, that handshake changing mid-process is stunning. Okay, so the acrosome-reacted sperm digests its way through the zona. It reaches the paravitallane space.
Right, that gap between the zona and the oocyte's actual membrane.
And finally, it fuses with the oocyte plasma membrane, the oolemma. You mentioned this fusion is integrin-mediated.
Yes, evidence suggests specific proteins called integrins on the oocyte membrane interact with ligands, like disintegrins, on the sperm membrane. So it's a specific cell adhesion and fusion event, not just bumping into each other.
Okay, fusion happens. One sperm gets through. But what stops more sperm getting through? Polyspermy would be disastrous.
Absolutely. The oocyte has a rapid and effective defense mechanism. Immediately following the fusion of the first sperm, the oocyte releases waves of calcium internally, which triggers the exocytosis, the dumping out of thousands of tiny cortical granules that were stored just beneath the oolemma.
They dump their contents into the paravetylene space.
Exactly. And the enzymes contained within these cortical granules cause two essential blocks to polyspermy.
Let's explain the physical effect of the first one, the zona reaction. How does releasing these granules actually harden the zona pellucida?
The cortical granule enzymes modify the structure of the zona pellucida proteins, particularly ZP2 and ZP3. They cleave ZP2 and chemically alter ZP3, effectively cross-linking the zona matrix, making it impenetrable to subsequent sperm. It also removes the binding sites for sperm, so the door is chemically locked and hardened.
Okay, that's the zona block. What's the second block, the oolemma block?
That's a change directly at the oocyte's cell membrane. The fusion event and calcium release also cause a rapid change in the electrical potential across the oolemma, making it refractory or unable to fuse with any other sperm that might have already penetrated the zona. This is a faster, but perhaps more transient block in mammals compared to the zona reaction.
So a double lock system.
Huh?
Fast electrical, then slower but permanent chemical structural lock.
Precisely. Ensures fertilization remains monospermic.
Now that successful sperm fusion also triggers something crucial inside the oocyte itself, doesn't it?
Yes. That calcium wave doesn't just trigger the cortical reaction, it also kickstarts the completion of the second meiotic division, which was arrested in metaphase 2.
Ah, right. Fertilization is the signal to finish meiosis.
Correct. This results in the extrusion of the second polar body into the paravetylene space, and the remaining set of female chromosomes decondenses to form the female pronucleus.
And the sperm's contribution. What actually enters the oocyte cytoplasm?
In humans, it's generally understood that both the head and the tail of the spermatozoon enter the cytoplasm. The sperm's plasma membrane gets left behind, fused with the oolemma. Inside the oocyte, the sperm nucleus rapidly decondenses, losing its tightly packed structure, and reorganizes to form the male pronucleus, carrying its 23 chromosomes, which could be 23X or 23Y.
So now we have two pronuclei, one male, one female, inside the oocyte cytoplasm.
Exactly. The ultimate outcome, the whole point, is the restoration of diploidy. These male and female pronuclei don't fuse immediately. They actually migrate towards the center of the cell. As they approach each other, their nuclear envelopes break down, and the chromosomes, one set from the mother, one from the father, align on the metaphase plate, ready for the first mitotic division.
So the zygote is technically formed when the chromosomes mix on that first spindle.
That's the moment. Diploidy, 46 chromosomes, is restored, and the unique genetic identity of the new individual is established. This single cell is the zygote. And of course, the sex is unambiguously determined at this point by whether the fertilizing sperm contributed an X or a Y chromosome.
XY for male, XX for female.
Correct. The sources even mention a possible immunological aspect here too, speculation about fertilizin on the ovum cortex reacting with antifertilizin on the sperm plasma membrane, suggesting another layer of molecular recognition.
Fascinating. Okay, the zygote is formed. Day 1. What happens next? Cleavage begins.
Yes, almost immediately. The zygote undergoes its first mitotic division, initiating the period of cleavage. This produces two daughter cells, called blastomers.
And this first division is slow.
Relatively slow, yes.
Yeah.
The two-cell stage is typically reached approximately 30 hours after fertilization. Remember, all this is happening as the zygote is slowly being transported down the fallopian tube towards the uterus.
And the blastomers keep dividing.
They do, by typical mitotic binary division. So 2 cells become 4, 4 become 8, 8 becomes 16, and so on. What's quite remarkable during this early cleavage stage is that the total volume of the cell mass does not increase.
Why not? They're dividing.
Because that rigid zona pellucida is still intact surrounding the dividing blastomeres. So the cells become progressively smaller with each division. They're partitioning the original large zygote cytoplasm.
Packing more cells into the same space.
Essentially, yes. By about 96 hours or 4 days after fertilization, the conceptus consists of a solid cluster of maybe 16 to 32 cells. It looks a bit like a mulberry, hence its name, the morula.
Morula. Okay, 4 days. And where is it now?
Around day 4, the morula, typically in the 16 to 64 cell stage, finally reaches and enters the uterine cavity, passing through the narrow uterine ostium, the opening from the tube.
And that transport down the tube was slow.
Deliberately slow, yes. Controlled by a combination of muscular contractions of the tube wall and the coordinated beating of cilia lining the tube. This slow pace is important. It ensures the morula arrives in the uterus at just the right time, when the endometrium is receptive.
And even within a morula, are the cells already starting to have different fates?
Yes. Even at this early stage, a crucial differentiation begins. The cells in the center of the morula tend to form the inner cell mass. These are destined to become the embryo proper. The cells on the periphery form the outer cell mass. These are fated to develop into the protective and nutritive membranes, primarily the placenta.
Okay, the morula has arrived in the uterine cavity. But it can't implant like this, can it? It needs to change form again. This is the transition to the blastocyst.
Exactly. This is the crucial prelude to actually embedding itself in the uterine lining. While the morula is floating free in the uterine cavity, this is around day 4 and 5, uterine fluid, mucus-rich in nutrients, starts to seep through those tiny canaliculi in the zona pellucida.
It gets inside.
It does. And this fluid begins to accumulate between the cells of the inner cell mass, coalescing to form a fluid-filled cavity. This cavity is called the blasticle or blastocyst cavity. As this cavity expands, it separates the cells into 2 distinct groups, transforming the solid morula into a hollow sphere, the blastocyst.
So the fluid pressure pushes the cells apart into layers.
Right. And this fluid accumulation also causes the blastocyst as a whole to enlarge. It starts stretching and thinning that surrounding zona pellucida. Eventually, the zona disappears. It's lased, broken down, likely by enzymes from the uterine lining or the blastocyst itself.
And this escape, this zona hatching, is absolutely required.
Absolutely mandatory for implantation. Without hatching, the trophectoderm, the outer layer of the blastocyst, cannot make direct contact with the endometrial cells to initiate the implantation process. The zona is a barrier that must be shed.
And the 2 cell groups are now clearly defined.
Yes. The differentiation that began in the morula is now solidified. The outer layer of cells forming the sphere becomes the trophectoderm. The cluster of cells pushed to one side. Inside the sphere is the inner cell mass. The sources mention this distinction is mediated by cell adhesion molecules like a chorion.
Trophectoderm outside, inner cell mass inside.
Correct. And their fates are now pretty much fixed. The trophectoderm is destined to become the chorion, the fetal part of the placenta. The inner cell mass holds those incredibly valuable pluripotent embryonic stem ES cells. They can become any cell type in the body, and they will form the entire future embryo.
Okay, hatched blastocyst floating in the uterus around day 5 or 6. Now comes implantation or nidation. You described this earlier as not passive, but a full-scale invasion.
It really is. It begins remarkably quickly, usually starting on the 6th day after fertilization. This typically corresponds to about the 20th day of a regular 28-day menstrual cycle.
And location matters.
Yes, it usually occurs in the endometrium lining the main body of the uterus, typically on the upper part of the posterior or anterior wall, near the fundus, not down near the cervix.
And it follows strict stages.
It does. The sources outline 4 key stages: apposition, adhesion, penetration, and invasion. But before any of this can happen, the endometrium itself must be ready.
It needs to be receptive.
Exactly. It must be in the mid-secretory phase, corresponding to days 20-24 of the cycle. This receptive state is primed by progesterone from the corpus luteum. The glands are secreting, the tissue is thick and vascular.
Okay, stage 1, apposition. First contact. How does that happen?
Apposition seems to involve the blastocyst, loosely associating with the endometrial lining. It's often mediated by these interesting structures called penopods.
Penopods?
Yeah, they're temporary, long, finger-like projections or microvilli that appear on the surface of the endometrial cells during the window of receptivity. They seem to perform a critical task. They absorb uterine fluid from the surface.
Why absorb the fluid?
Well, the uterine cavity isn't empty. It has fluid rich in glycogen and mucin, which provides some initial nutrition. But for the blastocyst to make firm contact, this fluid layer needs to be reduced or removed. So penopods might help draw the blastocyst closer by absorbing the intervening fluid. Close adhesion likely can't occur until this fluid is cleared.
Okay, fluid cleared, blastocyst is close. Stage 2, adhesion.
Adhesion is where it gets really molecular. It's a much firmer attachment mediated by a complex interplay of adhesion molecules on both the blastocyst, trophectoderm, and the endometrial epithelium. Molecules like integrins, selectins, and cadherins are definitely involved.
Like molecular Velcro?
Kind of, yeah. It's a specific molecular recognition process. And interestingly, it's primarily the polar trophoblast cells, those trophectoderm cells directly overlying the inner cell mass, that seem to be the most adhesive and invasive. They utilize specific factors mentioned in the sources, like p-selectin, heparin sulfate proteoglycans, and epidermal growth factor, EGF receptors, interacting with heparin-binding EGF on the endometrium.
So the embryo pole leads the way.
It seems so. And intriguingly, the signal for the trophoblast cells to really start multiplying and preparing for the next stage, invasion, appears to come from the inner cell mass itself. The embryo is essentially directing its own embedding process.
Amazing control. Okay, stage 3 and 4, penetration and invasion. This is the active burrowing.
This is where the trophectoderm really goes to work. The penetration occurs through the endometrial epithelial cells and then into the underlying stroma, often moving between the uterine glands. This is driven by the histolytic action enzyme-driven breakdown of the syncytiotrophoblast, which starts differentiating now.
Syncytiotrophoblast, a specific layer.
Yes, we'll detail it soon, but it's the outer invasive layer of the trophoblast. It secretes enzymes that digest the endometrial tissue, allowing the blastocyst to burrow deeper into the stratum compactum layer of the decidua, the specialized endometrium of pregnancy.
And this burrowing is deep.
In humans, yes. This leads to what's called interstitial implantation. The blastocyst doesn't just sit on the surface. It completely embeds itself within the endometrium, eventually becoming fully covered by it. This is different from superficial implantation seen in some other mammals.
As it burrows, the sources mention lacunae formation. What are those?
As the syncytiotrophoblast advances, vacuoles or empty spaces start appearing within it. These vacuoles fuse together to form larger, irregular cavities called lacunae. These become particularly evident at the embryonic pole, the leading edge of the invasion.
Are these important?
Critically important, because simultaneously, the advancing syncytiotrophoblast is eroding the walls of maternal capillaries and small blood vessels in the endometrium.
It breaks into the mother's blood supply.
Exactly. The syncytium penetrates right into the vessel endothelium. This allows maternal blood to seep out and fill these lacunae. This is the very beginning of the uteroplacental circulation.
And a major shift in nutrition.
A fundamental shift. Up until now, the conceptus was relying on stored reserves and uterine secretions. Now, with direct access to maternal blood in the lacunae, nutrition shifts entirely to an aerobic metabolic pathway, utilizing glucose and oxygen obtained directly from the mother's circulation.
But the invasion doesn't just continue indefinitely, does it? It has to stop somewhere.
No, it's highly regulated. Further penetration is actively stopped, likely by a combination of factors within the decidua itself and possibly maternal immunological responses acting as a brake. The original entry point in the endometrial surface gets sealed over, first by a fibrin clot, and later it's repaired by regenerating epithelial cells.
And when is this whole process, this interstitial implantation, complete?
It's remarkably quick. It's generally considered complete by the 10th or 11th day after fertilization. That corresponds to about day 24 or 25 counted from the last menstrual period, LM2. The blastocyst is now fully enclosed within the endometrium.
And this control invasion, this embedding, is governed by what? More molecular signals.
An incredibly complex dialogue. Endometrial receptivity itself is induced by progesterone, but specific molecules are key. The sources mention leukemia inhibitory factor, or LIF, which is crucial. Prostaglandins are involved, produced via the enzyme cyclooxygenase 2, or COX2.
And the immune system. How does the mother's body tolerate this invasion by genetically foreign tissue?
That's the million-dollar question, and it's where the complexity explodes. It involves profound immunomodulation. It's controlled by a whole orchestra of cytokines signaling molecules of the immune system. The text mentions interleukins 3, 4, 5, 6, 10, and 13. These generally promote tolerance rather than rejection.
So the immune system is actively managed.
Actively managed locally, yes, along with local peptides like epidermal growth factor, EGF, and insulin-like growth factor, IGF. And importantly, both the decidua, the maternal side, and the trophoblast, the embryonic side, are constantly synthesizing and responding to these molecules. It's a continuous molecular negotiation to allow invasion but prevent rejection or uncontrolled growth.
Okay, let's look more closely at the trophoblast itself during this. You mentioned it differentiates.
Yes, very early on, around day 7 or 8, that outer trophectoderm layer rapidly differentiates into two distinct layers. There's an inner layer of individual mononuclear cells with clear boundaries. This is the cytotrophoblast, sometimes called Langen's layer.
Cytotrophoblast.
And then there's an outer layer, which is a multinucleated mass of cytoplasm without distinct cell boundaries. This is the syncytiotrophoblast. Think of it like a giant invasive amoeba.
And the syncytotrophoblast comes from the cytotrophoblast.
Correct. The syncytium grows by fusion of the underlying cytotrophoblast cells into it. The cytotrophoblast cells are the proliferative stem cells.
And the syncytotrophoblast is the real engine of invasion.
It is. It's the layer that directly contacts maternal tissues, performs the enzymatic breakdown for invasion, forms the lacunae, and facilitates nutrient transfer. And critically, it's the primary site of hormone production very early on.
Like HCG, human chorionic gonadotropin.
Exactly. HCG synthesis by the syncytotrophoblast begins very early, by the 10th day or even slightly before. This is the hormone detected by pregnancy tests, and its crucial role is to signal the corpus luteum in the ovary to keep producing progesterone, maintaining the uterine lining and preventing menstruation.
So the embryo signals its presence hormonally almost immediately after implanting.
Instantly. And the cytotrophoblast cells themselves aren't uniform. The sources mention different specialized types. There's the villus cytotrophoblast, which lines the developing placental villi. Then there are extravillus cytotrophoblast cells.
Extravillus, outside the villi.
Yes. Some of these, the interstitial extravillus cytotrophoblast, invade deep into the decidua anchoring the placenta. Others, the intravascular extravillus cytotrophoblast, perform an absolutely crucial task. They invade the maternal spiral arteries in the decidua.
They invade the mother's arteries.
To remodel them, they replace the muscular contractile walls of these arteries with fibrinoid material, essentially converting them into wide, low-resistance channels. This ensures a massive, steady supply of maternal blood to the placental spaces later on. It's essential for fetal growth.
Wow. Okay, let's define the decidua properly. You said it's the pregnant endometrium. What's the decidual reaction?
The decidual reaction is the profound transformation that the endometrial stromal cells undergo in response to progesterone and later signals from the blastocyst. These normally small, spindle-shaped fibrous connective tissue cells enlarge dramatically, becoming polygonal epithelioid-like cells packed with glycogen and lipids. They're called decidual cells.
So the whole tissue changes structure.
The whole structure and function changes. The uterine glands also show increased coiling, dilatation, and intense secretory activity, pouring out nutrients. The whole endometrium becomes thick, vascular, and spongy, the decidua.
And does the decidua itself have layers?
Functionally, yes, it differentiates into 3 layers as pregnancy progresses. There's the superficial compact layer, or stratum compactum, right beneath the surface. This is a dense layer composed mainly of those decidual cells with the narrow necks of the uterine glands passing through. The blastocyst implants into this layer.
Okay, layer 1.
Beneath that is the intermediate spongy layer, or stratum spongiosum. This layer is much thicker and, as the name suggests, looser and spongy. It contains the dilated, tortuous bodies of the uterine glands and lots of blood vessels. This layer is clinically very important.
Why?
Because this spongy layer is the natural plane of cleavage, where the placenta separates from the uterine wall after the baby is delivered. The separation happens through this layer.
Okay. Critical for the 3rd stage of labor.
Exactly. And finally, the deepest layer is the thin basal layer, or stratum basal. This contains the blind-ending basal portions of the uterine glands. It's not shed during menstruation or delivery. Its crucial role is regeneration. The entire endometrium regrows from this basal layer after childbirth or menstruation.
So compact, spongy, basal. Got it. Now, after the blastocyst implants interstitially, covering itself completely, these decidual layers get renamed based on their location relative to the embryo, right?
That's right. They get specific names. The decidua basalis, also called decidua ceratina, is the portion of the decidua that lies underneath the implanted blastocyst, between the embryo and the muscular wall of the uterus, the myometrium. This decidua basalis will become the maternal component of the placenta.
The basalis base. Makes sense. What about the part covering it?
The thin superficial layer of decidua that covers over the top of the burrowing blastocyst, separating it from the main uterine cavity, is called the decidua capsularis, or decidua reflexa. Think of it like a capsule enclosing the conceptus.
Capsularis capsule. Okay.
And all the rest of the decidua lining the main uterine cavity, everywhere else that isn't touching the implanted conceptus, is called the decidua vera, or decidua parietalis, meaning wall.
Basalis capsularis was there. And what happens to the capsularis as the baby grows?
As the fetus and amniotic sac enlarge, the decidua capsularis gets stretched incredibly thin. Eventually, it comes into contact with the decidua vera on the opposite uterine wall. The potential space between them, the uterine cavity proper, gets obliterated. The capsularis and vera fuse together usually by about the 4th month of pregnancy.
So the decidua isn't just a passive bed. It's actively transformed, provides nutrition, forms part of the placenta, and critically, you mentioned it acts as that immunological brake, preventing the trophoblast invasion from going too far, right?
Absolutely essential role. It controls the depth of invasion, protecting the uterus from being completely consumed by the placenta, which can happen in abnormal conditions like placenta accreta. It's a dynamic, responsive maternal tissue. Right, so implantation is complete, the basic structure is set. Now we see rapid differentiation of both the supporting structures derived from the trophoblast and the inner cell mass itself. Let's start with the chorion.
Chorion, that's one of the fetal membranes.
It's the outermost of the two main fetal membranes, the chorion and the amnion. Structurally, the chorion consists of that outer trophoblast layer, both zygote and cytotrophoblast, plus an inner lining of primitive connective tissue called the extra-embryonic mesenchyme, which appears around day 9.
Trophoblast plus mesenchyme equals chorion.
Essentially, yes. And the key feature that develops from the chorion is the villi, the finger-like projections that form the placenta.
Okay, the chronology of villi development is important. It's a marker for embryonic age and placental development. How does it start?
It starts very early, around the beginning of the 3rd week after fertilization, so about day 14 to 15. We first see primary stem villi. These are basically just irregular finger-like projections of the syncytial trophoblast pushing outwards, but importantly, they have a core of the underlying cytotrophoblast cells.
So just trophoblast layers at this stage?
Yes, just syncytio and cytotrophoblast. These primary villi are poking into those lacunar spaces that are filled with maternal blood, which will eventually become the intervillous space of the placenta.
Okay, primary villi, what's next?
Around day 16, we get secondary villi. This happens when that extra-embryonic mesenchyme, the connective tissue layer lining the chorion, starts to grow into the central core of the primary villi.
So now it's syncytio, cyto, and a mesenchymal core.
Exactly. Syncytio trophoblast outside, cytotrophoblast layer underneath that, and now a core of mesenchyme inside.
And the final stage, tertiary villi.
That's the crucial step for function. This happens by about day 21, the end of the 3rd week. Within the mesenchymal core of the secondary villi, some of the mesodermal cells differentiate into blood cells and tiny blood vessels capillaries. This forms the villus capillary system.
The fetal blood supply within the villi.
Precisely. And this newly formed capillary network within the villi quickly connects up with the developing blood vessels in the main body of the embryo via the vessels forming in the body stalk, the future umbilical cord.
So by day 21, you have fetal blood flowing through capillaries inside the villi, bathed on the outside by maternal blood in the intervillous space.
That's the fundamental setup for placental exchange, established by day 21. This is why the fetal heart needs to start beating around day 21 or 22 to circulate blood through these newly formed villus capillaries.
Incredible timing. What about the trophoblast shell you mentioned?
Right. While the villi are developing, something else is happening at the tips of the anchoring villi, the ones that reach the decidua. Cytotrophoblast cells from the tips of these villi actually proliferate and penetrate through the syncytotrophoblast layer. They spread out laterally and fuse with cells from adjacent villi, forming a continuous layer of cytotrophoblast cells that sits directly against the maternal decidua basalis. This is the cytotrophoblastic shell.
So it forms a boundary layer between the fetal villi and the maternal decidua.
Exactly. It anchors the placenta firmly. The compact layer of the decidua immediately adjacent to this shell is sometimes called the trophosphere. And often, a layer of fibrinoid material called Niederbuck's membrane appears on the maternal side of the trophoblastic shell. It's thought to be a zone of fibrin deposition resulting from cell death and immunological interactions, possibly acting as another boundary-limiting excessive invasion.
Okay. Now, not all parts of the chorion develop equally, do they?
No. The fate of the chorionic villi depends entirely on their location relative to the decidua. The villi that are growing into the rich, thick decidua basalis underneath the embryo continue to grow, branch profusely, and become highly vascularized. This bushy, complex area is called the chorion frondosum. Frondosum means leafy or bushy. This will form the bulk of the functional placenta.
Chorion frondosum equals future placenta.
Right. Conversely, the villi associated with the decidua capsularis, the thin layer covering the embryo, find themselves in a much less favorable environment. As the embryo grows and the capsullaires get stretched and compressed against the decidua vera, the blood supply to these villi diminishes.
So they degenerate.
They gradually atrophy and disappear, mostly by the end of the 3rd month. This leaves the corresponding area of the chorion smooth and largely avascular. This part is called the chorion lave. Lave means smooth.
So the placenta ends up being a discoid structure because only the part facing the decidua basalis develops properly.
Precisely. The chorion frondosum persists and develops, while the chorion lave regresses.
Okay, that covers the supporting structures. Now let's zoom back in to the inner cell mass itself. What's happening there while the chorion is forming villi?
While all that's going on outside, the inner cell mass, or embryoblast, is also undergoing crucial organization. Around day 8, it differentiates into a 2-layered structure, the bilaminar germ disc.
Bilaminar 2 layers. What are they?
There's a dorsal layer, facing upwards towards the future amniotic cavity, consisting of tall columnar cells. This is the ectoderm. And there's a ventral layer, facing downwards towards the future yolk sac, consisting of smaller, flattened, or polyhedral cells. This is the endoderm.
Ectoderm and endoderm, the first 2 germ layers.
The very first 2. And this flat 2-layered disc is initially attached to the inside of the trophoblast, specifically the cytotrophoblast, by a stalk of extraembryonic mesenchyme called the connecting stalk. This stalk is vital. It will eventually develop into the umbilical cord, carrying the blood vessels to the placenta.
Okay, bilaminar disc established. What about the cavities that form around it?
2 critical fluid-filled cavities appear very early, defining the spaces around the germ disc. First, the amniotic cavity. This starts as a small space appearing within the embryoblast, or between the epiblast, early ectoderm, and the overlying cytotrophoblast around day 8, but it rapidly expands. By about day 12 post-ovulation, it's a distinct cavity dorsal to the ectoderm.
And the ectoderm forms the floor of this cavity.
Yes, the ectodermal layer forms the floor of the amniotic cavity. The roof is initially formed by cells called amnioblasts, derived likely from the cytotrophoblast or epiblast, which form the amnion membrane. This cavity fills with amniotic fluid, which will cushion and protect the developing embryo throughout gestation.
Okay, amniotic cavity on top. What forms underneath?
Ventral to the endoderm layer, the second cavity appears, the yolk sac. This initially forms as cells from the endoderm, hypoblast, migrate out to line the inner surface of the blastocyst cavity. This forms the primary yolk sac. It gets remodeled slightly later into the secondary yolk sac, often just called the yolk sac.
And the yolk sac isn't really about yolk in humans, is it? That's its function?
No, not like in birds or reptiles. In humans, the secondary yolk sac has several crucial early functions. It's the initial site for fetal hematopoiesis. The very first blood cells are formed in its walls. It's also involved in the synthesis of alpha-fetoprotein, AFP, an important fetal protein. And its endodermal lining is considered a very important zone for nutrient and molecular transfer from the mother, via the coalomic fluid initially, to the developing embryonic gut.
So amniotic cavity above the disc, yolk sac below. What about the space around them?
Right. Remember the blastocyst cavity, the space between the inner cell mass and the trophectoderm. As the extraembryonic mesenchyme fills this space, small cystic spaces, or lacunae, start appearing within this mesenchyme itself. These small spaces rapidly fuse together to form a large new cavity called the extraembryonic coilum, EEC, or chorionic cavity.
So another cavity forms outside the amnion on the yolk sac, but inside the chorion.
Exactly. This EEC surrounds the amnion and yolk sac, effectively separating them from the outer chorionic wall, the trophoblast lined by mesenchyme, except at one crucial point, the connecting stalk, which traverses the EEC to link the embryonic disc to the chorion.
And what happens to this extraembryonic coilum?
Early on, it's quite prominent. You can often see it on early ultrasound scans as the space around the yolk sac and amniotic sac. However, as the amniotic cavity expands dramatically with the growing embryo inside, it pushes outwards. The EEC gets progressively smaller and is gradually obliterated, usually by the end of the 12th week.
How is it obliterated?
The expanding amnion membrane eventually fuses with the mesenchyme lining the inside of the chorion. So the EEC disappears, and you're left with the fused amniochorion membrane, the familiar bag of waters.
Okay, so we have the bilaminar disc sitting between the amnion and yolk sac, suspended by the connecting stalk within the chorion. But 2 layers isn't enough to build a body. We need 3.
Precisely. The next major morphological change, and arguably the most profound organizational event in early development, is gastrulation. This is the process that establishes the 3rd germ layer, the mesoderm, and transforms the bilaminar disc into a trilaminar embryonic disc.
And when does gastrulation happen?
It begins around day 14-15, marking the start of the third week. It starts with the appearance of the primitive streak.
The primitive streak, what is that?
It's a visible thickening, a groove, that appears along the midline of the ectoderm, specifically the epiblast layer, at the caudal tail end of the embryonic disc. It's formed by the proliferation and inward migration of epiblast cells.
Cells are moving inwards.
Yes. Cells from the epiblast migrate towards the primitive streak, detach and move downwards, invaginating or pushing between the existing epiblast layer, which we now definitively call the ectoderm, and the underlying hypoblast layer, which we now definitively call the endoderm.
And these migrating cells form the third layer.
Exactly. These cells, spreading out laterally and cranially towards the head end, between the ectoderm and endoderm, form the intraembryonic mesoderm. This establishes the crucial three-layered structure.
Ectoderm, mesoderm, endoderm. The trilaminar embryonic disc.
That's it. Gastrulation sets up the fundamental body plan. From this three-layered structure, all the tissues and organs of the body will eventually develop.
And this marks the beginning of the embryonic stage proper.
Yes. The period from the start of the third week, gastrulation, up to the end of the 8th week is considered the embryonic period. This is when organogenesis, the formation of organs, occurs. Individual organ differentiation begins rapidly once the 3 germ layers are established. By the end of the 8th week, the major organ systems are all laid down, and the embryo is morphologically distinguishable as human.
Okay, let's quickly review the derivatives of these 3 fundamental layers. They form an organizational map for everything that follows. Ectoderm.
The ectodermal layer, the outermost layer, primarily gives rise to structures that maintain contact with the outside world. So the central and peripheral nervous systems, brain, spinal cord, nerves, the sensory epithelia, like in the eye and ear, and the epidermis, the outer layer of skin and its appendages, hair, nails, sweat glands. Also the pituitary gland, subcutaneous glands, mammary glands, and the enamel of teeth, plus the mucous lining of the nasal and oral cavities.
Okay. Middle layer.
The mesodermal layer is responsible for the structural components, movement, and circulation. So it forms bones, cartilage, all types of muscle, skeletal, smooth, cardiac, the cardiovascular system, heart, blood vessels, blood cells, the lymphatic system, the kidneys, the gonads, ovaries, and testes, the suprarenal adrenal glands, and the spleen. Also, most of the genital tract and the serous membranes lining the body cavities, pleura, pericardium, peritoneum, the mesothelium.
It's a lot. Finally, the innermost layer, endoderm.
The endodermal layer primarily forms the epithelial linings of the internal tracts and associated glands. So the epithelial lining of the entire gastrointestinal tract from pharynx to rectum, the liver, the gallbladder, and the pancreas. Also the epithelial lining of the respiratory tract, trachea, bronchi, lungs, and most of the mucous membrane lining the urinary bladder and the urethra, as well as the thyroid, parathyroid, and thymus glands. Hashtag outro.
Wow. Okay. We have really charted a course there. We've completed, I think, a truly detailed map of just the first 2 weeks of life. But the timeline is so incredibly tight, it almost demands this level of mastery to appreciate it.
It really does.
Just think of the clockwork precision we've outlined. The 2-cell stage around 30 hours, the morula entering the uterus at 96 hours, that complex interstitial implantation completed by day 10 or 11.
And those crucial vascularized tertiary villi, the real start of the placenta, in place by day 21, which is exactly when the fetal heart needs to start beating to make use of them.
The synchronization is just breathtaking. If we step back, as you said earlier, the central theme has to be that synchronization, hasn't it? Between the hormonal signals that LH-FSS surge, the rise in progesterone, and the incredibly intricate cellular responses.
Absolutely. Things like capacitation having to happen at the right time, the instantaneous zonal reaction to block polyspermy, the transformation of the endometrium into the decidua. It's all perfectly timed. This whole process is just a biological masterpiece of coordinated invasion, communication, and control.
And what struck me again is seeing the sheer complexity of the molecular signaling that governs that invasion phase during implantation. The number of regulatory molecules involved, LIF, the COX2, all those interleukins and growth factors we listed, it really shows that implantation isn't just burrowing. It's arguably the body's most sensitive diplomatic mission.
That's a great analogy. A full cabinet of cytokines is needed just to negotiate and maintain the peace between the genetically distinct mother and embryo.
And having this detailed mastery-level knowledge isn't just academic, is it? It's the absolute foundation for understanding reproductive health, pregnancy complications, and of course, reproductive technology.
Definitely. Think about in vitro fertilization, IVF. especially intracytoplasmic sperm injection, or ICSI, where a single sperm is injected directly into the oocyte.
Right, you bypass the natural journey and the zona pellucida barriers.
Exactly. You manually bypass the complex ZP3-ZP2 binding mechanism, the corona radiata challenge, the whole acrosome reaction sequence needed to penetrate the zona. But even though you inject it, the sperm still needs to be appropriately prepared or activated, sometimes chemically, in the lab to mimic aspects of capacitation. Its nucleus needs to decondense properly, and it needs to trigger the oocyte activation sequence correctly for the pronuclei to form and development to proceed. The lab techniques have to respect and, where possible, replicate the natural choreography we've discussed.
So now that you, our listener, have this detailed map, this mastery-level understanding of these first 2 weeks, let's leave you with that final provocative thought we touched upon earlier. It really is a biological tightrope walk, isn't it? Implantation.
It truly is.
The maternal immunological system must somehow be precisely modulated, suppressed just enough locally to allow the invasion of the blastocyst carrying paternal antigens half foreign tissue. Yet, simultaneously, the decidua must act as the vigilant brake, utilizing those chemical signals of physical layers we talked about to ensure that the invasion stops at exactly the right depth, before it goes too deep and damages the uterus.
It is this moment of necessary, highly controlled immunological paradox, a sacrifice of normal defense mechanisms, but only just enough and only where needed. It's absolutely essential for the survival and success of the pregnancy, a constant balancing act from the very first week.
A profound biological negotiation. Thank you for joining us on this deep dive.